Thank you. I have to say, I think it's my luck to arrange my oral after the recognition, because today's talk is first established from his group to use the anatomy landmark to align human and macaque. Today, I'm going to present from functional perspective how I'm going to use the functional data to help with alignment between species. So let's start with Dr. De Rensen's work. As you can see here, he proposed a set of anatomy landmarks to try to align macaque, monkey, and human brain, and it aligns anatomy, a few unimodal anatomy landmarks very well, but a large proportion of social cortex leave it limited with a landmark, so we don't know how accurate those regions can be aligned based on the anatomy landmarks here. But this pioneer work has been first created, the surface expansion map has been informative for look at the evolutionary route to compare to, for example, the childhood cortical expansion, individual difference variation, and the local or distance functional connectivity to understand the functional organization across the species. And recently, this cross-species alignment work has been done from the Roger Marsh group from Oxford, and using the wet matter tracks to create the wet matter landmarks and to map the connectivity, wet matter connectivity between the vertex to the landmarks region to compare cross-species, and which he referred as a blueprint. Another one is to try to use the chimpanzee mining map as a midpoint to map the human brain and the macaque brain together. From functional perspective, Dante Martini proposed this movie paradigm by playing the same movie to macaque, monkey, and human, and trying to align the monkey brain and human brain by the activity fluctuation where they are watching the movie. But as you can see here, those methods were limited in a awake monkey and also limited in a visual cortex as well. So the question becomes to how are you going to align the human and the macaque based on functional data? So if we look at this evolutionary tree, we find out there are few anchored regions, which is primary visual, auditory, empty regions, and the sensory motor regions are quite common, not only in primate, but also all the mammals as well. So if we look at this white regions, which is associated cortex, and from different perspectives, let's say all the associated cortex is gathering all the information from the primary sensory cortex, and if we think about this hierarchy, and only matters difference between species, that is how large this associated cortex between species. And in rodents, primates, this associated cortex may be small, but in human may be large. But in terms of the functional information flow, this hierarchy from the unimodal regions to heteromodal regions is similar. So this model has been proposed in 1998 and recently been demonstrated based on the fMRI data from Daniel Margulis, and showing these one gradients spanning from the unimodal region, visual sensory modal, to the deformal leg region. Another gradient is spanning across different modality, visual and sensory motor regions. And this is consistent in human and macaque. So this motivates us, maybe we can make use of this gradient to align human and macaque together. So here we use the human data from SAP dataset and macaque data from recent open resource primate data exchange. All of these data are all available online. And this is what we do. As you can see, the bottom left, that is a joint similarity matrix we proposed to put the two species together. The diagonal matrix, which is a within species connectivity, voxel-wise, and the off-diagonal matrix, which is the connectivity, the similarity profile based on the connectivity to the landmarks regions. When we put this joint matrix together, we propose this joint embedding, which is the same algorithm as Daniel Margulis did, and we can simultaneously extract the gradients, which is matched between human and macaque. And here I just visualize the first and second dimension. The first dimension is a gradient across different modality, visual and the sensory model. And second gradient is the gradient spanning from unimodal to the heteromodal regions. As you can see here, I visualize these landmark regions. It's in this functional space, the landmark regions will have a relatively low distance. And we can also project those functional gradients back to the surface. As you can see here, the first, second gradients are already visualized in the functional space. But from the third gradient to the other gradients, it reflects the anatomy landmarks as well. But one advantage of this method is it's not only focused on landmarks, but it creates a continuous map, can cover the entire brain, and offered us a quite informative features can align a human and macaque. So we put all of this on the sphere and trying to use the most recent surface matching algorithm, multimodal surface matching, and we're able to match the human brain and macaque brain together and all the other way. So this is a validation. Remember, all the features are functional features, and no anatomy features, no curvature has been used in this MSM. So we are able to map the human Malin map back onto the macaque surface, and you see how consistent this human to macaque Malin map to the Malin map we extracted from the macaque directly. So next thing is, if we think about this topology, think about MSM, we project everything onto a surface, that we constrain the topology between macaque and human. But this might not be true, so we propose this functional homology index, which is for each vertex on the human brain, we find the matched vertex based on MSM. But we create a searchlight and search what is the maximum similarity between species for this vertex. And we assign the maximum similarity back onto the human surface. And this suggests the most likelihood we can find in macaque that is homology with the human, this vertex. So this is functional homology map in human brain looks like. It captures the high homology regions capture the landmarks as well. But I want to point out one specific region, which is FFA regions here. Remember, this FFA regions is not included in the landmark, but the functional homology mapping able to capture the border of the FFA regions, which is consistent with Matthew Glance's postulation very well. I also mapped this onto the flat map. As you can see here, it's reflected the diversity of the functional homology on the cortex. And if we test it for the different network, we see this hierarchy. Basically, the unimodal regions, visual and sensory modal regions, showing the high homology index, and the frontoparietal and deformable network regions showing the low functional homology. If we zoomed in this into the visual cortex and the sensory motor cortex, and there's, I have to point out 2013, Buckner proposed this tethering hypothesis, which suggests that all of those anchored regions are serve as anchor, and all the associated cortex regions, just the regions start to emerge between the anchored regions. So this early visual hierarchy, ventral visual hierarchy, and empty regions, all the X is ordered by the distance from the anchored region, V1, or empty regions, to the far neighboring regions. So you see all of those homology index shows this hierarchy consistent with this early visual, ventral visual, and the distance to the empty regions very well. And another thing is from at the sensory motor cortex, this order is from the dorsal to the ventral part. And if you think about the dorsal to ventral part represents the function, the dorsal part is more about our hands and body, and the ventral part is about our eyes, face, and tongues, more related to the language or emotions, so it has the least functional homology or cross-species similarity here. And let's zoom in to the associated cortex, which is the frontoparietal and attention network. Recent study from Buckner's group suggests even in the frontoparietal and attention network, there are subsystems. For example, the frontoparietal network A more related to the trigeminal network, and the frontoparietal network B is more related to the attention network. So we see this network A and network B has the different functional homology as well. And in general, attention network has a higher homology than the frontoparietal network. Then if we move to the default network, we can consider this meta-analysis-based subsystem. There suggests there are three systems. One yellow one, that's a core system related to our self-reference, and the blue one, that is the medial dorsolateral system related to our theory of mind of the mentalizing personal social stuff. This is and the blue one, the green one, that is the more related to the memory. And you see the core part, which is ourself, is less likely have the homology index. And if we break into the details and we see the importance of deformed network has correlated with the functional homology index as well, which suggests that the more important deformed subregions has the least functional homology, which suggests the more important deformed regions emerge later in the evolution. And if we seed these regions in macaque and search around the whole human cortex, find which regions share the most similar deformed configuration with human, and we see the VMPFC, DMPFC, and hippocampus regions were most likely as a human deformed network, and all the other PCC and anchorage areas doesn't show the same patterns, which suggests human macaque monkey may have the deformed-like regions, but it's on the way to develop as a human does. It doesn't have the similar level as human deformed network. So we also compare this to the cognitive function and see all of those basic functional.
Could you please work towards the conclusion soon?
Okay.
Thank you.
Sorry. To wrap up, it's just we demonstrate this joint embedding can map the human brain and macaque brain together and develop this functional homology, which suggests the hierarchy and supports the tethering hypothesis. And it also suggests that the so-called deformed network might not be different, might not be the same in monkey brains. And I would like to thank for all the colleagues and the fantastic collaborators. Thank you.